Okay, welcome back again. Welcome back in the ongoing video lecture series of blood group, and this is, I think, going to be the part 6 or 5th part of the blood group lecture series. So, today I am going to elaborate the topic that I have given a very brief description in, I think, the second or first video in the blood group video lecture series. Where I have told you about the blood transfusion. That means, suppose you have a blood group containing individual and you want to donate your blood group to your loved ones. So, on the basis of his or her blood group, it will be determined that is you have the ability to give your blood to him or her to save her life. Okay, so for this blood transfusion is done. So today I am going to talk about this in very brief. Okay, so here two individuals will be in picture. One is donor who want to donate, another one is recipient who received the donor blood. Okay, so here we need to bothered about the donor's RBC, and now you need to tell you now you need to ask me why we are only bothered about donor RBC, because donor's blood will be the foreign particles, and we all know antigen is present on the surface of RBC of donor blood group. And in recipient we need to talk about him or her serum, because in serum he or she have their antibodies. Okay, so in the overall process is called type and cross match. I have written this already, type and cross match test. So first of all we need to type the ABO blood type or RIG status. That means what antigens are present on the surface of RBC. Now if you want to know today about how these antigens are originated on the surface of RBC, then you can easily go back into my YouTube channel and find out the biochemical basis of blood group. Okay, and then come back to this and it will be more helpful to you. Okay, so we need to find out the antigens that are present on the surface of RBC. Okay, now one thing, remember one thing, that RBC not only have those antigens, only A and B and Rh antigen, it have many antigens. Okay, that are not usually typed. Now, if somehow this serum contain some antibody against those untyped antigens, it can be a problem. Okay. So, as there are no typing here to the machinery or mechanism or theory is available, so before transfusing the donor's blood to the recipient blood, we need to perform a test, that is what we call cross match test. In that test, we need to take the donor's RBC and recipient serum and mixed it in a test tube. Okay, and after that we need to examine with the microscope. If the proposed transfusion is incompatible, that means what? That means the recipient serum has antibody that can interact with RBC's some antigens. Okay, if it is, then we can say the proposed blood transfusion can result in the formation of agglutination and can be dangerous to recipient serum. So that is the overall idea of type and cross match. Now we are going to do a question on this that was asked in 2016 December CSIR for more clarity. So here is the question. So let's try to read what they are telling us. If in a blood transfusion type A donor blood, please highlight this thing. Type A donor blood means we need to bother about A antigen. Okay, as I have previously told you that in donor's case we need to bother about the antigen present in that particular individual is given to a recipient having type B blood. You also need to mark this as B blood group containing individuals is the recipient. So we need to talk about anti A antibody. Okay, we need to talk about anti A antibody. The red blood cell of donor blood would agglutinate, but the recipient RBC would less affected. They are telling us that the red blood cell of donor blood is agglutinated, but the recipient RBC would least affected, and that is the normal case. As donor blood RBC is the foreign particle, as it has our antigen. Okay, but the recipient RBC would be less affected. This observation can be explained in the following statement, and you need to talk. You need to tell them that what statements are incorrect. Okay, so let's read the first statement. They are telling us that agglutinin in recipient plasma could cause agglutination by binding with type A agglutinogen. So remember, I have told you that, or I am again told, I am teaching you, okay, that antibodies that agglutinates antigens are called agglutinins. You need to remember this thing. And similarly, antibodies those have the ability to precipitate antigen will be referred to as precipitins. Okay, you won't need to bother about this here, because blood group is totally about agglutination. There is no place for precipitation. Now, in agglutinins in recipient's plasma cause agglutination by binding with type A agglutinogens. The agglutinogens are in turn referred to as antigens, and there is no term exists in referring to as precipitogens. You need to you might wonder about this, as in case of agglutination antigens referred to as agglutinogens, so in case of precipitation there will be something like precipitogens. It is not that so. Okay, so they are simply they tell us that agglutinin, that is the antibody in recipient's plasma, caused agglutination by binding with type A agglutinogens. So this is absolutely correct, as donor blood group have type A. Okay, that means they have A antigen on the surface of themselves. Okay, so that is the reason why type A antigen should be agglutinated with the help of recipient plasma. Now let's move to the next one. Welcome back again. So in the statement B, they are telling us that the agglutinins of donor blood was diluted in recipient plasma, resulting in low agglutination. Now remember one thing, that you are a donor and your blood is transfused into recipient serum. So your blood serum will be simply agglutinated in recipient's body. Okay, because it is agglutinated. No, sorry, simply diluted in recipient's body. Because why? Your blood is very small amount, it will be simply diluted in recipient's body. It is the normal thing. Okay, but that does not mean that it will the result of low agglutination, because agglutination is only going on the donor, the antibody donor antigens, donor antigens and recipients antibody. Okay, so the agglutination of donor blood was diluted recipients plasma, resulting in low agglutination. That can be a reason, but we are not bothered about that, because we are only focusing on donors antigen and recipients antibody. Okay, in low so it is a correct statement, but you know, don't need to bother about this in many questions. Okay, you only need to find out donor antigens and recipients antibody. That is the whole thing. So in statement C, they are telling us low titer of anti-A agglutinin. Anti-A means it is coming from B blood group. Remember that is the cause of low agglutination of recipients RBC. Anti-B diluted. Okay. So remember one thing, that our recipient was B. Okay. Its recipient is having the type B blood. That means it has B antigen and anti A antibody. So there is no place for anti B antibodies for performing in agglutination, as we are only bothered about RBC's antibody, and RBC's antibody here is anti B. Okay, so anti, we are not won't need to talk about that, because anti A antibody is simply coming from what? Low titer of antibody is the cause of low agglutination of recipients RBC. No, this is not that case, because the recipient's RBC will be agglutinated by type S antibody, that is anti B, and it is diluted. Obviously it is diluted, but here for this statement you need to remember the term that I have told you, that your blood do not make any kind of antibody against your own antigen. Okay, your antibody will not show any type of response against your own antigen. So that is the idea. So it will be again a wrong statement, and a high agglutination of donor RBC is a outcome of high titer of anti-B agglutinin. So high titer of anti-B agglutinin means our recipient is B, so the recipient's antibody will be A. So high agglutination of donor RBC is the cause of higher titer of anti-B agglutinin. This is also wrong because we are only talk about anti-A antibody. So our correct answer will be 4, that is C and D, because they are telling us only incorrect statement to find out. So I hope this video will be helpful for you, and if it is, please like the video, share with your friends and subscribe my channel. Thank you.